Hi everyone, we're Group 17 and we'll be talking about the study of positive feedback regulation of microglial glucose metabolism by histone H4 lysine 12 lactylation in Alzheimer's diseases based on research done on lab mice. Alzheimer's disease, the most prevalent neurodegenerative diseases with symptoms such as cognitive decline and memory loss. Microglia, the brain's immune cells, are pivotal in AD pathogenesis, as the increased lactate production and inflammation when shifted from a oxidative phosphorylation to glycolysis under stress. The result of the study uncovers a positive feedback loop involving histone lactylation in microglia that drives AD progression by enhancing glycolysis. This loop exacerbates microglial dysfunction. However, by inhibiting PKM2, the positive feedback loop is shut down, which results in a shift from glycolysis back to oxidative phosphorylation, thus improving memory and cognitive decline. These findings suggest that targeting the glycolysis and the positive feedback loop could be a compromising therapeutic approach for Alzheimer's diseases.
To investigate the role of PKM2 in microglial cells in Alzheimer's disease mice, the research team carried out 3 key experiments. First is Western blotting, a technique that involves extracting proteins from cells, separating them by size via jaw electrophoresis, and then using specific antibodies to detect the target protein. This method is used to obtain PKM2 protein levels in microglial cells from 6-month-old WT and 5XFAD mice. Second is lactate assay, which uses the lactate colorimetric assay kit that converts lactate into a product that can produce a color change and thus measured spectrophotometrically to determine lactate concentration. Third method involves breeding 2 strains of genetically modified mice to produce a new strain of mice and knocking out the PKM2 gene in microglial. This allowed researchers to analyze the effect of PKM2 deletion in the microglial cells of Alzheimer's disease model mice.
Figure 3a shows the results of a Western blot analysis, which measures the protein levels of PKM2 in isolated microglia from wild-type and 5XFAD mice. Beta tubulin here is used as a loading control, and the results show that the 5XFAD mice have a much higher levels of PKM2 protein due to the increased band intensity, which indicates an upregulation of PKM2. Whereas figure 3b shows the intracellular lactate levels in microglia isolated from the wild type and 5XFAD mice. The bar graph shows that 5XFAD microglia have a much higher lactate levels than the wild type. Each dot here on the graph represents 1 mouse with 4 wild type mice and 5 5XFAD mice. The increased lactate levels in the 5XFAD microglia match the results obtained from figure 3a, where there were a higher PKM2 levels seen. This is statistically significant as indicated by the 2 stars. This indicates that the microglia in the Alzheimer's disease mice have undergone metabolic reprogramming, resulting in increased glycolytic activity and lactate production.
As seen on the lactate levels between Alzheimer's disease mice with and without PKM2 protein, the mice without the PKM2 showed dimmer bars on the Western blot for both pan-KLA and H4K12LA, meaning these 2 glycolytic genes are less expressed. Thus, the lower H4K12LA and pan-KLA is related to the lower lactate levels in the PKM2 knockout group, indicates that removing PKM2 will lower the usage of glycolysis. This provides evidence that a shift from glycolysis back to oxidative phosphorylation took place in the mice microglia, because lower lysine lactylation products indicates lower levels of lactate and suggests lower usage of glycolysis. On the Western blot, histone H4 is compared between 2 groups to help ensure that equal amounts of histone proteins are loaded across different samples, which is crucial for making accurate comparisons between samples.
In conclusion, rats with Alzheimer's showed higher levels of lactate, meaning more use of glycolysis within the microglia, indicating more cognitive decline. Furthermore, inhibition of PKM2 causes a decrease in lactate levels, indicating the reduction of glycolysis, thus increasing oxidative phosphorylation, which supports the hypothesis of a metabolic pathway switch in the microglia as part of the cause to Alzheimer's. This study proposes the idea of reducing glycolysis in the microglia to reduce the cognitive impairment, which can be a potential pathway for Alzheimer's treatment in the future. However, this idea awaits further testing in human microglial cells for more accurate results. Thank you for listening to our presentation.